HIV, because HIV basically works the same way. So we use this word lysogenic, where, and my example is a little confusing up here. So it says lysogenic or temperate. You can use those words interchangeably. And we're talking about viruses that infect, but then after they infect, they can kind of sit there in an inactive state for some time. So I'm going to show you a picture of what I mean by this. Now for the viruses that we just talked about that kill, that have that lytic effect, they have that killing effect. I gave you examples on Monday. I said they're referred to as T2, T4, T6. My example for a bacteriophage that infects lysogenically is called bacteriophage lambda. And this virus works differently because you're going to see in the picture it infects, but then it kind of sits there for a while before it kind of gets reactivated later on. So yeah, so this idea of latency, viruses can do this, yeah.
So in the meantime, is it just multiplying, or is it just
the virus isn't multiplying, but the bacteria can multiply.
Oh.
Yeah, and then during that binary fission, the viral information kind of just gets passed down.
So the bacteriophage is also infecting the new bacteria.
Yeah, like the information's already in there. Yeah. But since the host doesn't die, it just does its binary fission. Wow. So I'm gonna show you a picture. What I want to do is like ignore the top part, because the top part is what we already talked about. The top part is what we mentioned on Monday already. So that killing effect, that lytic effect, we already went over that piece of it, the attachment, entry, biosynthesis. Everything is assembled, and then they burst out. That's what I showed you in the picture. What I'm introducing that is new is on the bottom, this idea of latency. So we kind of start off the same way. The virus attaches, it enters. I'll show you a picture. It enters. But the difference is, instead of the virus shooting right away to biosynthesis, where it starts copying itself right away, instead what ends up happening is the genetic information kind of slips into the host, genetic information, and the two kind of meld together, fuse together. And then the genetic information of the virus just kind of sits there quietly. And we call this kind of latent state when that happens, when the genetic information kind of slips in and just kind of sitting there and nothing's really happening. We call this a prophage state. So that's kind of what the question that came up. So the viral information it's in the host, but nothing is happening with it yet. It's just kind of sitting there. The bacteria is totally fine. It doesn't lyse, open, and die. And it just grows normally. It does its binary fission like it normally does. But every single time that bacteria splits, that information from the virus gets passed down, right? So every 30 minutes as it divides, that information gets passed. And then unfortunately that can have bad consequences for us, because sometimes this DNA can code for some really horrible things like toxins. So I'm going to give you an example. All right. Okay, I know this figure is, it looks super, super, super crazy, but I just want to follow from like the beginning. Basically, this is a picture format of everything we talked about. So the picture over here was basically a summary of the picture we already went through, where the virus attaches. See there, it's kind of squatting down, the DNA enters, biosynthesis, and then they burst open and they're released. We call that the lytic cycle because the host actually lyses. But there are a lot of other viruses out there that don't kill right away. Instead if we start at the beginning, instead they attach, the DNA enters, but instead of going right into biosynthesis, we don't do that yet. The DNA basically kind of slips into the DNA of the host, so it kind of slips in there, and they kind of integrate and become one, and the bacteria is totally fine, it's just kind of hanging out undergoing binary fission, nothing happens to the bacteria. And then eventually, usually there's some sort of trigger, there's some sort of stressor for the host. And then this virus kind of all of a sudden gets the activation signal to go ahead and shoot into biosynthesis. And then eventually we get that machinery going, and then they all burst out, and we eventually end up here at the very, very end. But in the meantime, we kind of have this whole process called latency. So we refer to that as like an inactive state, right? So the host is totally fine. It's a little bit of a problem though, because we have viral information though getting passed down through binary fission, and sometimes that information can code for not so bad things. And so with this lysogeny life cycle that we're talking about, it's very significant because, as a reminder, viruses, they do kind of look like little aliens, and they're kind of like little transporters. And in a way, viruses can shuttle information around. So that does happen, right? They don't have a lot of DNA or a lot of RNA, but the little bit that they do have sometimes can code for things like toxins, and that information can get shuffled around. So in a way, you can kind of view viruses as little transporters of genes, because they are. They basically have genetic information packaged in this little protein shell, and they kind of hop from host to host, and they move that information around. So this is definitely, you can kind of see viruses as another way that genetic information moves around. We've talked about, like conjugation, that's one way. Plasmids can move around from bacteria to bacteria, but also viruses. Viruses can definitely come into the picture and also move information around. And then the other bad thing is, we said, since some viruses can infect quietly, and that information can kind of integrate into the host, like literally become part of the host, sometimes that information will carry some the blueprints for things like toxins. So this is saying when a little virus actually integrates into the host, a lot of times it brings new information to the bacteria, and the bacteria might gain the ability to make something like a toxin. So that happens too. So the information for toxins doesn't necessarily have to come from a plasmid. It could come also from a viral infection. And we're talking about bacteria. This is applying really to bacteria, not for us. But the reason why this is so bad for us is because now we're saying that there's this whole world where viruses can infect bacteria, and then unfortunately those bacteria can infect us, right? So we can kind of get like these super bacteria that are carrying around these toxins. And this happens all the time. I only want to give you 2 examples. Your textbook has like a long list of examples, but I only want to mention 2. There's 2 bacteria where actually this has already been known to happen. This has already occurred. Clostridium botulinum and that Vibrio cholera, remember that bacteria that causes cholera? That really severe form of diarrhea. Those 2 bacteria, that's already happened to. They got infected by a virus, by a bacteriophage, and that virus brought in information for a toxin. And so now these 2 bacteria are able to make this very, very potent toxin, and that's basically what makes us sick, right? So these bacteria, the only reason why they're able to make their toxin is because they got infected by a virus themselves, right? So we're going to talk about Vibrio cholera and Clostridium botulinum. We're going to talk about the toxins that they make. And we actually commercialize the toxin made by Clostridium botulinum. Does anybody recognize from the name? Botox, yeah. So we're going to talk about Botox. So when I say toxin, that's actually the toxin made by Clostridium botulinum. So we've made a lot of money off of Botox. All right, so yeah, so there is a lot of significance there. Well, what I want to do though is kind of transition. We have kind of the outline though for how viruses work, but I want to focus in on the animal viruses, which are the viruses that infect us and animals, so humans and animals. And we're going to start off with, again, attachment. So we're going to go through this step. Step number 1 is always attachment, always. So in order for a virus to infect us or an animal or a bacteria, it always has to attach. For attachment, sometimes that attachment can be really specific. So we're going to talk about this with HIV. There's a lot of viruses that only infect the liver. There's viruses that only infect your T cells, right? Or there's viruses that only infect your respiratory tract, like the flu virus. For that attachment process, so one of the things we said kind of in the beginning of this lecture is that viruses are either enveloped or naked. If you're naked, you don't have that envelope, and so you just kind of use your protein shell called the capsid. That's what you're kind of using to attach. In my picture though, these two are pictures of enveloped viruses, and in the enveloped viruses, what makes them a little bit different is usually they have these protrusions called spikes that kind of come out, radiate out. And the envelope viruses usually use those spikes to do that attachment step. So it's really, really important for that attachment. So attachment, we're saying the spikes, the spike proteins, that's what usually the virus uses to attach to its host. And a lot of times those spikes can be really specific, really specific. We're going to talk about flu and kind of highlight that with the flu virus because it works the same way. For the flu virus also has spikes surrounding the virus, and that's what it uses to attach to our airway, our respiratory tract. So I kind of mentioned this too on Monday that knowing what the spike or the capsid likes to attach to is really important because from like a pharmaceutical point of view, if there's some way that you can interrupt this attachment process, that could potentially prevent that virus from causing an infection, right? So sometimes it works and sometimes it doesn't. We're going to talk about with HIV why it doesn't work out. Entry. There's always an entry, and I'm going to show you a picture for the entry. The thing that I wanted to emphasize here, though, is the viruses that infect us enter a little bit differently from those bacteriophage. The bacteriophage, I said, they kind of do the squatting, and they have the lysozyme, and they just kind of thread the DNA through. For viruses that infect us, it's basically these two mechanisms. One's called pinocytosis. Pinocytosis is basically, we're talking about very small things that are engulfed by the host. So our cells can take up things from their environment. And pinocytosis, what that means, is kind of a fancy word, but all that means is that it should be entire. The entire virus is actually taken up into the cell. So I'll show you a picture. It's almost like a little pouch forms, and it grabs the whole virus, and the whole virus just kind of sinks in. So that happens a lot. We call it pinocytosis. The whole virus enters. But sometimes, not necessarily the whole virus has to enter. Remember, it's just the genetic information that really has to enter, not necessarily the protein code or the spikes and everything else. It's the genetic information. So the other popular entry route is called fusion. And fusion is a little bit different because what ends up happening is the envelope or the capsid actually melts, kind of blends in with our cell membrane that the virus is trying to get into. And then all that enters is the really like the genetic information or the capsid with the genetic information. So not the whole virus enters when it's fusion. And then usually there's an uncoding step. Uncoding just means, let's say the whole virus does actually get in. Let's say it's everything, the spikes, the envelope, everything. Then the virus has to go through an extra step called uncoding. You got to strip all of that away because, again, we're trying to get to the DNA or the RNA. That's what the virus wants to get into its host, right? So that's why this says the DNA or the RNA is finally released. Everything's stripped away, peeled off, and released into the host. So we call that uncoding. Then the rest is basically the same, biosynthesis, right? The virus gets busy, starts to replicate all of the parts. So animal viruses, just like phage though, there might be a delay between 3 and 4 because again, if it's kind of that late, it decides to do the latent route. Not all viruses have to jump to number 4 right away. Sometimes there can be a big lag. And then maturation, everything's assembled, that's the same. So all the different parts are assembled together. So the outline is basically very similar to viruses that infect bacteria, but the release is a little bit different. So at the end we have release. Viruses that infect us, they can also lyse and burst out just like the bacteriophage, how we had in the picture for the bacteriophage. But not all viruses have to kill their hosts when they get released at the end. We're going to talk about HIV because HIV does A, called budding. What HIV does, instead of killing the host right away, it just slowly buds out. And when we say bud, what the virus kind of does is it kind of has little docking stations up at the cell membrane, and it kind of just pinches off. So there's no, dramatic, like bursting open type of release. They just kind of slowly pinch off and slowly get released from the host that way. When they kind of pinch themselves off, they basically pick up a little bit of your cell membrane. They pick up a little bit of your cell membrane, and that's what it uses for its envelope. So remember that third layer that we said is the envelope? They're getting their envelope from you. So they're like, oh, this is awesome. I don't have to make my own envelope. I'll just grab it from you as I kind of slowly bud out. I'll just steal a little bit of your membrane, and then that serves as the envelope. So yeah, these are the 2 release mechanisms. Everything kind of in between and the outline is basically the same. There's just some subtle details, differences between phage and, yeah.
So is that why the white blood cells can't get it or attack it because it has our membrane?
Yeah, that's another problem. It's kind of like a cloaking. Yeah, it kind of cloaks itself in us. It's like our material. Kind of like another sneaky.
It's nature's little ass.
So I don't want to get into like every single detail behind penocytosis and fusion. I just wanted to show you in the middle. When I use that word penocytosis, what I mean is that the whole virus actually enters your cell. So there's the virus, it attaches to you, and it kind of sinks in a little pouch, and then the whole virus enters the host cell. On the top, fusion is a little bit different because fusion, a virus attaches, there it is kind of attaching, and then what ends up happening is after it attaches, it triggers this fusion event, and fusion kind of sounds like we have 2 things kind of melding into each other. So in this case, it's that envelope which kind of sinks in with your membrane. And the only thing that enters is the capsid with the genetic information. Eventually, though, we move to uncoding. Remember we said, so that protein capsid is like stripped away, and then the genetic information enters. So the final goal is we always want to get to the genetic information. The genetic information, that's what the virus cares about actually getting in. So that's what I mean by penocytosis versus fusion. And then I actually have a picture too. because, there we go, the budding is kind of a better picture of the budding. Because the budding, I said, the virus kind of forms like a little docking station on the top. This is actually your cell. So this would be your cell. And then the virus is already infected, and it went through all the steps, and everything's all assembled. And then it kind of cruises up to the top, and it forms like a little bud. And then it just slowly pinches out, and it grabs a little bit of your cell membrane, that's your cell membrane, and it kind of buds out, it gets released. And then in that membrane, it'll have all the spikes. So the virus actually delivers all the spikes to the top of your membrane, and then it kind of buds out. And then we have our fully formed virus here at the end. So we call that budding. All right, so my last couple things that I'm going to mention is, as far as the duration of a viral infection, definitely you can get some differences. As far as the effect of a virus on its host, right? There are what we call acute viral infections. And acute viral infections mean that the infection process is rather short or pretty brief, right? So we use kind of the common cold as a good example. So just the common cold virus that moves around. Usually the symptoms develop really fast, and then you recover pretty quickly thereafter, right? Not all viral infections are acute, though. It'd be nice if they were all acute, but a lot of them can be very persistent. And when we use that word persistent, it means that your immune system actually does not clear out the virus in a short period of time. The virus can remain in your tissue and linger over as long as many, many years, actually. This can theoretically happen with the measles virus. So that was kind of the amusing part when we had, of course, measles made big headlines when we had that whole debacle at Disneyland. And it's very amusing because a lot of people, especially like the older generation, they're like, I got measles, it was fine, wasn't a big deal. But what a lot of people don't know is that although it is pretty rare, it can happen. One of the complications of the measles viral infection is in some people, the virus can actually persist in the brain tissue. And so one of the complications here is the subacute sclerosing panencephalitis. So many years later, the virus can reawaken and the virus can start becoming active again and actually cause inflammation of the brain, and it can be deadly. So that is something that can happen with the measles. Now also another example of a persistent type of infection. So number 3 is also persistent. When we say something is chronic, that means again the virus is just lingering. It doesn't go away like an acute infection. The difference though between persistent and chronic is in a chronic infection, that virus is always active. Always active. It's always in that biosynthesis step. In the persistent infection that we talked about with measles, the virus is not in that biosynthesis stage all the time. It kind of sits there quietly and then later on, yeah, it's more of that latent. Versus chronic, like the classical hepatitis B, people that get Hep B, and that virus is active. So it's active in your liver, it's always being released, and that's why you get that potential complication with liver cancer, because the virus is always active. Now there is like also another type of latent infection where we use that word latent, and this is also persistent. So 2, 3, and 4 are kind of like different types of persistent infections. What makes the latent a little bit different though is the virus also sits quietly in an inactive state in your body, but there's a trigger. There's usually some trigger that causes it to reawaken, and then it disappears again. And then that trigger comes back, and it reawakens, and it disappears. So you kind of get like these spikes where you get sick again, you recover. You get sick again, you recover. And the classical example is herpes, good old herpes virus, right? Usually what's that trigger that causes the virus to reawaken for herpes? Yeah, stress is the big one, right? So you either stress or like some change to your immune system, or maybe you're on medication or whatever it is, but your immune system status changes or you get really stressed, the virus starts multiplying, and then your immune system clears it out, and then it kind of quiets back down, but it never goes away because it's persistent. And then you get that trigger again, and then it just comes back. So the cold sore comes back again and again and again, right? So those are kind of the major effects that different viruses can have on us. And then the last thing that I'm going to mention is the link between viruses and cancer, because there is a link there for sure. Luckily, though, viruses are not responsible for like the majority of cancer cases, which is good. Cancer, though, is definitely one of those complex diseases, very complicated. And there's many different reasons why a person would develop abnormal cellular growth. So we refer to tumors as collections of cells that have grown abnormally. And then, of course, we have different terms for describing how far that's gone with the abnormal cellular growth. So for benign tumors, those cells stay within a very defined area. And then we use that word malignancy when those cells actually start to leave the primary site and actually start spreading. Cancer, just like HIV, those two diseases are they're really challenging things to work on. There are a lot of labs across the country that just study this, metastasis. Metastasis is the spreading process. So there actually are dedicated labs where they're just studying how cancer cells spread. But yeah, there's kind of a lot that goes into this. And it's challenging because no tumor cell is like the other. It's kind of like with HIV, we're going to talk about that too. There are genes that come into play though with cancer. So we do know that for some people there's definitely a strong genetic link. Sometimes it runs through families, but also sometimes these genes can be carried around through viruses. And we use a special word called oncogenes. Oncogenes are the abnormal genes that actually code that tell your cells to divide. So we call them oncogenes. And those are when the genes actually become abnormal, in an abnormal state. All right. So my last word that I'm introducing is this. When we say that a normal cell becomes cancerous, like that transformation from normal to becoming cancerous, we call that transformation. A cell goes through this process. It's usually a process where it's transformed into an abnormal cell. And there's a lot of different reasons why some cells can become abnormal, right? So again, it might just be genetics. So that information maybe was already built in your DNA. It might be environmental, right? We know of some things, some strong links, right, like smoking or whatever it is. And then we also know that viruses can come into play. Viruses contribute because viruses can actually carry oncogenes. Those abnormal genes that tell your cells, all right, divide like crazy. Viruses can carry around sometimes that information. And if you get infected, then with that virus that's carrying those abnormal genetic information, it'll trigger your cells to start dividing like crazy. So that can happen. So there are what we call cancer-causing viruses out there, oncogenic viruses. Those do exist. There's not a lot of them, but there are a lot of them, some out there. And then the last one is my viruses. They can pick up, so there's a world of oncogenic viruses, and then there's also kind of just inadvertently, since we just said viruses are little shuttles that can shuttle information around, sometimes they pick up just the normal genes. They're not abnormal. They're normal, but they just accidentally pick them up. It's on accident, and they go and they infect you, and they still trigger your cells to divide like crazy. So that can happen too. So what we're kind of emphasizing is that since viruses shuttle around from host cell to host cell, they can definitely transport the genetic information for cellular division as well. All right, and one of the things we're going to mention at the end of the semester is HPV, human papillomavirus. And what cancer is that linked to? HPV. Cervical, yeah. So there's cervical cancer. I have just like the three examples of oncogenic viruses. But yeah, HPV definitely disrupts our cellular divisions. So in some women that get infected, the virus will disrupt that process and inadvertently trigger the cervical cells to kind of divide. And divide and divide. And so my three examples are HPV, which we're going to talk about at kind of the end of the semester. HPV is a virus that loves to infect mucosal surfaces. So the primary concern is cervical cancer, but what people forget is men can also develop cancer from HPV. It's pretty rare, but it can happen. So they can develop penile cancer, or anal. And then hepatitis B, there's a strong link with liver cancer. Hepatitis loves to infect the liver, and usually hepatitis infections are always chronic. So they're always undergoing biosynthesis the entire time it's in your body. It never goes latent. And so since it's so active and undergoing biosynthesis, sometimes in that chronic process, it'll actually cause damage to your cells and trigger the development of cancer. And then there are leukemia viruses that we know of called HTLV, and those can infect like blood cells, like the T cells. All right, so those are my three. And I have a percentage, so 10 to 15% of cancers are caused by virus. So it does take up a slice of the pie. Probably, though, stronger indicators would be genetics and environmental reasons. Okay. So we're done with chapter 6. We're just going to start to introduce kind of the HIV lecture. What I am going to do too, before I forget, is the two kind of things that we emphasize with the entry and the penocytosis and fusion and budding. On course sites, I'm gonna actually post like short videos of those three things. You can, and then there's like a short little online quiz. I don't like grade them or anything, but the system will ask you some questions. So I'll post that, and you can kind of test yourself on those three things. But where we go? Here we go. Okay. So we're just going to start this section, and this section is only dedicated to HIV and prions. On your syllabus it says chapter 25, but it is not the entire chapter. So it's literally like the first section only of 25, so we're not covering the whole chapter. But we are going to mention a little bit about those prions again, because prions get included in the discussion for micro because they are very small infectious material. They're not alive and they're not cells. But what makes prions very different is the fact that they're non-living, but they're also not viruses. So they're just strictly made of protein. So prions are infectious proteins. They're not even made of cells. And what makes them really interesting is for the longest time, people thought that they were viruses. They were like, prions have to be viruses. And then we realized that we had it all wrong, and they're actually not viruses. They're just made of protein. So these prion proteins, they don't have the capsid or any of the machinery associated with the virus. And what makes them really unique is that they're really hard to denature. Because really, like the majority of proteins out there, you can denature them no problem or heat them up. But these are super resilient, so it's really difficult to denature them. They can be denatured in something like an autoclave, but it takes a lot longer. It doesn't happen in 15 minutes. So I have a picture of the man who's responsible for discovering prions, and actually his last name kind of sounds like prion a little bit, Prusner. Stanley Prusner, which sounds like prion a little bit. And he's actually an American scientist, and he was given the Nobel Prize for his discovery of prions. And this happened in the late 90s, so it actually wasn't that long ago, although it feels like a long time ago, for his discovery of prions. And what fascinated him, what got him interested was in the mid-70s, he was a resident working up north in a hospital. And a female patient was admitted, and the female patient was in her 30s, and she had signs of dementia. And dementia is not something that you find in younger people. So he was fascinated by this and thought there must be a reason why this patient in her 30s is showing signs of dementia. So he got interested and he started studying this. And he actually, in the process, became quite a big laughing stock because people thought he was wasting his time because they said it has to be a virus. It's just a virus, so just let it go. And he persisted and actually uncovered that they're actually prions, abnormal prions. So I have a picture of, since prions are just proteins, you guys know that proteins, they have helical beta sheets in them. I have a picture of the normal and abnormal forms of a protein. So on top, this is like kind of the shorthand. The acronyms here are used to designate the normal and the abnormal forms. On the top it says PRP. C, and PRPC is the quote-unquote normal form. What PRPC stands for is it stands for prion protein, and then cellular, that's the normal form. And what makes this totally interesting is everybody has a normal form in their body. So we all have it. It actually coats our brain and spinal cord.
In the membrane?
Yeah, in the membrane. Yep. And on the bottom, I have a picture of the abnormal form called PRPSC. And PRPSC is the abnormal form. Or you can kind of think of it as like the infectious form. So we got the abnormal form / infectious. And it stands for prion protein, not cellular, but the SC stands for scrapie. SC, there we go. Stands for scrapie. And scrapie is the name of the disease that sheep get. So it's the prion disease found in sheep. Found in sheep. The reason why they call it scrapie is because when they develop their prion disease, it affects their brains, and it triggers them, it's really strange, it triggers them to look for anything to scrape against, and they start basically like scraping themselves to death. So they call it scrape B. So it definitely alters the brain, and it does the same thing in humans as well. It alters our brains. So the last thing I'm going to mention about the prion diseases is it's not just humans that get prion diseases, animals get them too. Sheep, cows, also things like deer, elk, they can get prion diseases. And it's all caused by the abnormal form of the protein. It's just a protein, but it's folded differently. That's what makes it definitely different from a virus. And it can trigger brain damage. And in humans, usually this takes a while. It's kind of definitely a long process for that damage to develop, many years. But the set of diseases that prions cause, they're called transmissible spongiform encephalopathies. And we've used that term in chapter 1. And remember that descriptor spongiform describes what happens to the brain, the changes to the brain, because it basically starts to look like a sponge. What ends up happening is these abnormal proteins, they start building up in your brain. So your brain and spinal cord, they start transforming into the abnormal form, and then it starts to form these collections, and that's why your brain looks like a sponge. And this also happens in other animals as well. All right, so how would we pick up a prion disease? Like where would we get these abnormal prion proteins from?
Eating
eating. So my last thing that I'm going to mention is, one of the ways is definitely ingestion of a food product like the beef, where we had the mad cow outbreak. That's definitely one way that prion proteins can move around. So that's the last thing. I just want to see how they're transmitted. So we can acquire them through ingesting the abnormal forms. Luckily, though, eating the abnormal form, like in beef product, it can happen, but luckily it tends to be rare, so that's good. But it can happen. Another thing to think about too is surgical equipment. Surgical equipment that comes into contact with brain or spinal cord. You want to make sure all of that material is sterilized between patients, because that could potentially be something that comes into play. But a more common thing is it actually can be hereditary. So it can get passed down. If your parents are carrying a copy of the abnormal form, that can actually get passed down. You can actually inherit the abnormal form. So it's another thing from parents. Yeah.
Either parent?
From either parent.
Wow.
Yeah. That's rare, but it can happen. That's more common than eating the through food. It's for it to be hereditary. All right. And then we said, unfortunately, there's no way for anybody to diagnose that you actually have a prion disease. The only way that you would know is upon autopsy. So they actually have to look at your brain to know for sure.
But that's after you're done.
After you're dead. I know. Yeah. All right. Okay, so all right, so we're done mentioning prions. We're just going to introduce a little bit about the HIV.
Are there like specific like environment, climate, or region where they thrive at, or?
Yeah, you know what? So I was just going to say that because I have ingestion, like eating contaminated beef or eating something that's got abnormal prions in it. And they're the most notorious place in the world is like Papua New Guinea, because Papua New Guinea, there's kind of like a tribe that lives there, and it was a part of their ritual that when somebody would pass away, they would eat their brain. And so prion diseases are actually really prevalent there because of that ritual.
That's the hereditary.
They're hereditary, yeah. So yeah, since they've eliminated that process, then transmission has been cut down. But yeah. So eating human brain tissues, I didn't put it on there, but that's another definite way. It can move around.
So zombies.
Zombies, yeah.
Zombie protein.
All right.
Iron and Prions, maybe that's what.
Yeah, zombies. Alright, so we're going to mention HIV. We've come a long way with understanding HIV. Not so much in the coming up with a cure, but the natural host. So what was interesting is there was like a big kind of official scientific study. So in the world of science nerds, like what we have are basically different scientific journals that research studies are published in. And the two like top-notch journals, one's called Science, so that one's an easy one to remember, and the other one's called Nature. And those two, these are basically journals. They're like the most competitive journals to get your studies actually published in. In Nature, there was a group of scientists that came out with the results of their study, and they actually declared that they figured out what the original host of the HIV virus is. And it's a particular chimp in Africa. Now, in last Monday, I said there's an HIV-1 and an HIV-2. So HIV-1, which is the one that really affects the majority of the world, originated from a chimp. HIV-2 came from not a chimp, but kind of a related they call it an old world ape. So it's not a chimp, but it's related to the chimp. And this study came out in 1999. So it wasn't until 1999 that they officially came out and said, all right, we know what the original host of HIV virus is. And it's a chimpanzee from West Africa.
A family of chimps or one particular chimp?
Oh, definitely, probably like a family. Yeah. So we've known that in chimps, so definitely way before 1999, there are viruses that run through chimps that are similar looking to HIV, and they've been around for a long time. And in this study, they finally basically figured out that the first human exposure was definitely probably a hunter. So one thing that we don't have to worry about in Long Beach is we don't hunt for our meat. But in other parts of the world, it's very common. It still happens even now and today. We're going to talk about this later on. But there's what's called bushmeat hunters. So people hunt chimps, basically, and they use them for food. And there's a lot of blood exchange, usually in that hunting process. So as they're slaughtering the chimp, using it for food, usually blood is exchanged. And that's basically what the study came out and said is that's how HIV jumped from the chimp into the first human. But these viruses have been running around through chimps for a long time. It was just a matter of time before they actually made their way into us. And of course, the first cases in the United States started showing up kind of in the mid to late 70s. And then people started realizing, so it was really mostly like New York that was predominantly the city where these cases started to arise. And of course, there was a big stigma with HIV/AIDS as being linked as like the gay disease, right? Or the homosexual disease. But if there's anything that we know about any virus, it doesn't matter if it's HIV or any other viruses. Viruses don't see race or color or anything like that. They just want a host. So they don't care. They don't care about sexual orientation. They just want a host. So for our background, HIV belongs to a family of viruses called Retroviridae. And the genus is called lentivirus. And there's kind of a variety of lentiviruses out there. And AIDS, we do know, it's been established that HIV is the cause of AIDS. And AIDS is an acronym that stands for Acquired Immunodeficiency Syndrome. And that was first reported in the 80s here. But definitely, so another part of this too is people are always interested in the viruses that move through animals, because we're going to talk about that. It's always a concern that another virus, this could happen to us again, where a virus moves from an animal to the human population. Maybe doesn't cause something like AIDS, but something equally, as dramatic or bad. And definitely education has helped. So when a virus first kind of reared up in the United States, we didn't know how it moved around. Now we know a lot about transmission and how the virus actually infects. So the number has slowed down a little bit, but still, we do have a lot of new infections every year. What we're going to do is kind of take a look at, not so much the numbers, but the demographics, how the demographic has shifted a lot with HIV. One thing we know for sure is that males make up more than half of new infections than females. So really predominantly, as far as newly diagnosed people, it's mostly males. All right. But the thing that I wanted to share with you is our demographics. Our demographics have definitely shifted a lot. So what I have is I grabbed it from 2011. It's a little bit old, but what I think is interesting is the CDC and a lot of the kind of health organizations are trying to think of more specific ads for targeting certain populations and getting education out, right? So on the right, what they did was they kind of did a very rough sample of, we did like a ethnic breakdown for the population of the United States. Unfortunately, the categories are really broad, so it's like Asians are just Asian, and then we have all sorts of Hispanic, Latino just lumped in together. But anyways, when we look at the pie, it says 63% of the population in the U.S. are considered white, 17% is Hispanic, 12% African American, and then we have Asian and Indian, Alaska Native, which is a little bit more specific. On the left, though, is the pie chart for diagnosis. So new HIV infections for 2011, and we can see that on the right, while African Americans only make up 12% of the population, they made up 47% of the newly diagnosed HIV infections. So while, Hispanic Latinos made up 17%, they made up 21% of the pie here. And even though whites are taking up 63%, they made up a smaller percentage of the newly diagnosed HIV infections. So definitely our demographics have shifted. And one of the things they're always working on is targeting specific populations to kind of get education out so that people know they can have ways of protecting themselves, right? All right. So we now know that HIV doesn't just infect homosexuals. It can infect anybody. So the next big segment is, how does HIV infect? You guys probably know a lot about this by now. But how would you pick up an HIV infection?
Dirty needle.
Through what?
Dirty needle.
Yeah, dirty needle can definitely be in there. What else?
Blood.
Yeah. So because of the blood, ignore my red. Because of the blood, sexual contact is always in there. So in parentheses, I put, what does sex mean? Just to be like super clear. Because yeah.
Sorry.
Yeah. So just to be clear, we're talking about all forms of sex. So definitely vaginal is in there. Anal is in there, yes.
Oral.
And technically oral. Yep. All three. While oral is not a high percentage, you have to include it because theoretically, yes, the virus can move through bodily exchange of fluids through oral sex. It's not very common, but it can happen, so we have to include that under when we say sex. Needles are in there as well, definitely. Not sterile needles, contaminated needles, so we don't have to be freaked out with the normal sterile needle, but we were talking about reusing needles, right? So usually most cities support those anonymous exchange programs that they have for people where they can dump off used needles and get fresh ones. There was one city up north that got a lot of flack because they got rid of that program. They were like, we don't want this program around. We don't want people dumping off their needles. And the rates of HIV transmission obviously went up in that city. Bodily fluids, yes. What I'm going to do is give you technically every single bodily fluid. Blood, really like blood is the main bodily fluid that we get concerned about. Technically, so, I have pus and pleural fluid. Those are really, 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 really rare. It's just technically I included every possibility. Really blood is the primary bodily fluid. Pus is not really a concern usually. Yeah.
Semen?
Yes, semen, yes. Semen, and one thing that people forget about is breast milk. Yeah, the virus does move through breast milk. So for HIV positive moms, they cannot breastfeed because the virus can pass through. Mother to child, yes. So the mother to child transmission can happen during pregnancy. It can happen during labor, and it can also happen post-pregnancy through breast milk. So yes, vertical, we call that vertical transmission. And the rate of transmission is about 30%. So the chances of that happening. My 30%, that is if mom is not on any HIV meds. That's without any treatment, no treatment.
How much farther,
So,
how much lower is it?
When mom is on meds, it drops to like around 8%. Yeah. Which is better. Yeah.
Not great.
Yeah, not great, but it's better. And then also, I always like to share the statistic too with the needle sticks, because I know a lot of you guys are pre-nursing. I grabbed it from the Center for Disease Control website. What they did was they analyzed the percentage of healthcare workers, nurses, physicians, people who picked up HIV or hepatitis B from a needle stick because you're on the job. And the rate of HIV transmission was less than 1% amongst healthcare workers. For hepatitis B, though, it was 30%. So one of the things that I wanted to emphasize, because we said there's naked and envelope viruses, HIV is an envelope virus, and usually envelope viruses are really wimpy outside the host. The naked ones are actually a lot more persistent outside the host, and hepatitis is actually in that naked category. They're naked viruses. So actually, hepatitis is kind of the hepatitis B. At least we have a vaccine for B, but C. People get more nervous about hepatitis C and B versus HIV, at least from needle sticks. All right. So high-risk groups. We're saying anybody who's high risk, that would be intravenous drug users. I had to update that because I just had drug user, and I was like, wait a minute, I need to be more specific. Intravenous drug users. Obviously, unprotected sexual contact. The reason why I put especially male to male is because the CDC actually broke down, oral sex, male to male, female to male, and all the different ways, the different partners. And the highest rate of transmission, definitely so far, is male to male. So, but unprotected sex, doesn't matter with who. Mother to child, yes, an unborn fetus. And now, number 4, blood transfusion. I specified the dates because really now, blood transfusion now in 2016 would not put you really at high risk for acquiring HIV. That was a higher risk like in the 80s though, in the 80s. So, and it was really interesting because the LA Times did this big article about, the people who donate blood. Because they have that ban that, I didn't know this, that Like homosexuals couldn't, I don't think they could donate, right? Donate blood. And they had this big, huge article in the LA Times about, okay, it's time to lift the ban. You know, the virus moves around all sorts of people, so it doesn't matter. And on kind of the other side, they interviewed people who picked up their HIV infections from a blood transfusion, and there are like these horrible stories, of course. You know, they were in hospital, and they got their blood transfusions, and they were tainted with HIV, and now they're infected for the rest of their lives. And some of these people were like, no, keep all the bans in place. I'm like, okay. So it's kind of an interesting dichotomy there. So the last thing that we're going to do is kind of go over the structure of HIV. So at least the good news for HIV is it does not do well outside of the human host or animal host. So that's the good news. They've tried to do studies on this. It's really hard, but the studies that they have done, they basically like calculated how long the virus can exist outside of the human body. And they say on average, it kind of does okay for about 6 hours, and then eventually it just kind of, I can't say dies, but it deteriorates, breaks down. Definitely drying of blood and bodily fluids, just the drying process helps to inactivate the virus, so that's good. Still, you know, we want to take precautions though, and we want to protect ourselves. So any situation where we have exchange of, especially blood, right? Blood is kind of the main bodily fluid that we have to be concerned about. But at least that is the good thing. At least HIV is not in that naked category, because the naked viruses can actually persist a lot longer on an object. So what we're going to do is kind of break down what the virus actually looks like the different parts, the different components of the virus. And we're going to kind of sketch this out on the board together. I notice your textbook actually has the best picture for HIV, better than any of the other better than any of the other textbooks that I know of. And the reason why is because they literally label like every single possible part. All right. Okay, so in your textbook you have kind of a better picture. I'll draw like my own little skimpy picture here inside. Alright, so, and then yes. But the picture in your textbook is nice because they literally labeled, especially on the inside, every sort of possibility as far as the enzymes that are carried in there. But what's nice is when we label the different parts of the virus, then when we talk about how the virus infects, you'll know like which part of the virus is doing what. So HIV is definitely an envelope virus. It does have an envelope, that extra layer that covers the capsid. The capsid is that protein coat on the inside with all the circles, like capsid. The beginning of chapter 6, we said capsids are either like icosahedral shaped, helical, or complex. This capsid falls in that complex. It doesn't really, it's not icosahedral and it's not helical. Kind of has its own unique shape. And then there are spikes, but the big deal about the spikes is, and I noticed the LA Times, they like to call them lollipops. But the outside, which we call the head here, is made up of one protein, and the protein is actually labeled the same way on the picture. The protein's called GP120. So that's just the name of the protein that makes up the little head of the spike. And then there's a different protein that actually makes up the stem of the spike, and we actually call that GP41. So GP is kind of a shorthand for saying glycoprotein. So it's kind of made of sugar and protein. So 120 is one type of protein and 41 is a different protein. Those are really important because we're going to talk about what the parts of that virus, how they contribute to the infection. On the inside, I have the two squiggly lines because these are my two RNA pieces. HIV is an RNA virus. It's an RNA virus. And it only has two strands of RNA, that's it, which is fascinating. So only those two pieces of RNA. And then inside there are a variety of enzymes, that's labeled in the picture. One of the enzymes that we're going to kind of make a big deal about, one of the enzymes is called reverse transcriptase. And reverse transcriptase is probably one of the most significant enzymes that we're going to come back to and talk about. There's another enzyme that the virus packages. It's called integrase. And integrase kind of sounds like integration, which is what the virus does. So we're going to talk about that. And then there's one last enzyme called protease. And those are the three most important enzymes: reverse transcriptase, integrase, and protease. But only two pieces of RNA, so that's interesting, is enough to cause all that damage inside of us. All right. So what we're going to do, we're not going to have time to finish this all, but for the last thing today, we're just going to start off by mentioning the whole process of the HIV infection starts with attachment, right? So this is step number one right here, the spikes. The whole process starts when the virus, so the virus will enter either through an abrasion or a cut, or we have that sexual contact. And once it makes its way through your mucosal surface, it hits your blood right away. And right away, it's the spike that mediates that first step, which is the attachment step. So that's kind of the last thing that we're going to emphasize. GP120 doesn't just bind to anything, it loves to bind to your white blood cells. So it does have like a very specific host in your body. That GP120 spike that we had on the board loves to bind and attach to not any white blood cell, but in particular your T cell. And we have different types of T cells, but there's one type of T cell in particular that it loves to bind to, and that's your CD4. We call them helper T cells. It's the helper T cells. They're the ones that HIV loves to attach to. It has a very high affinity for that type of cell. So the way this works is our white blood cells are also coated in proteins. We're going to get to talking about T cells later on in the semester. What makes it really confusing about immunology is they always use letters and numbers for describing names of proteins. So it makes it really confusing. Because you see this random letter number, CD4, and you're like, what is that? But CD4 is just the name of a protein that's on top of that T cell. So CD4 helper T cells, they have that particular type of protein. Another type of T cell would have a different protein designated with another letter and number, so it gets very confusing. I remember when I was in college and I was working in a lab, and it was an immunology lab, and they'd always be like, hey, okay, we're going to have this meeting and talk about
what's going on in our lab group, and everybody was talking in letters and numbers. They're like, the CXCR4 binds to the GP1, blah, blah, and then that triggers IL-2, and then IL-4 gets made, and then IL-12 gets upregulated. And I was sitting there, I was like, what are you talking about? So it makes it really, really confusing. But, so that's the one protein that you find on top of the helper T cells. Now, the thing is you can kind of think of the CD4 protein being like the main kind of docking station for the virus. There's also an additional helper protein, though, that the virus likes to make contact with. We call those co-receptors. And there's two particular co-receptors that the virus loves to attach to. These are just more proteins. So I'm going to show you a picture. These are just additional proteins that the virus has to make contact with before it enters. One of the proteins is called CCR5, and the other protein is called CXCR4. These are the two other proteins that the virus also docks with before it actually enters. Now, what's totally fascinating, I was talking with a student about this, is that there are a small percentage of people that inherit a bad copy of their CCR5 protein. So from mom and dad passed down to them both the information for the bad copy of CCR5. What that means for those people is that they are naturally resistant to HIV. They can never get HIV because their protein is misfolded and HIV cannot dock on top of their T cells. So that's another kind of interesting, obviously people have a strong interest in studying those people. And it's a very small percentage of Europeans that carry that mutations, like less than 1% of them are in that special group. And so that's kind of an interesting thing because we do know now that, recently, right, they came up that they think that the second person has been cured of HIV, but basically so far, for sure, we know there's only been one person cured of his HIV infection, and it was done on accident, but what they did was they gave him a blood transfusion, and the blood that they took and they gave to him came from a person that had this CCR5 mutation, and he was actually cured of his HIV. That's only one for sure case out of all the cases out there. And they call him the Berlin patient. He's got like a special descriptor. But yeah, so those proteins are really important. I have a picture, just so you can see back here. Here's those spikes, the GP120. And what it's doing is it's making contact with those proteins on the top of our T cell. So this is supposed to be the T cell down here. And on our T cell, we said there's one protein called CD4, so it kind of has that picture there. And then usually there's an additional protein like the CCR5, and it's the two together that kind of grab onto the GP120. Once those proteins kind of make contact and are kind of docked in there, then it triggers the virus to start entering your T cell. So now that we've had contact, entry is the next step. And the entry is driven by the stem of the spike, not the ball, but that GP41. So remember in our picture we had labeled the stem as GP41. The GP41 will then act like a drill. It's kind of interesting. So it literally kind of pokes through and drills through your T cell, and then the virus enters by fusion. So the envelope kind of melds in with your T cell, and what actually enters is the capsid and the two pieces of RNA, and then all those enzymes that we had written on the picture. So the whole virus actually doesn't enter, but it's the capsid and the genetic information and the enzymes that actually enter. All right. So entry is triggered by GP41. Once we've entered, the last thing is that enzyme reverse transcriptase. Oh, I think what I'll do is, yeah, I'll just end on showing the picture here. So yeah, the reason why I said that we know for sure one person's been cured is because I don't know if you guys saw recently, they think that possibly another person has been cured. You see that? That just came out like literally last week or something, very recently. But also in Great Britain. It was a patient in Great Britain, and they think that they cured him of his HIV infection. It was kind of sad. We did have that case in Long Beach that was like a while back, where an HIV positive mom transmitted it to her child. This was in Long Beach. And the doctors treated the baby, and they actually thought that they had cured the baby of its HIV infection. That actually turned out to be false. So that was kind of, that was really sad. But here we have the attachment picture. So here's like all the docking stations. And CD4 is really like the main docking station for that GP120. So they kind of make contact. And then usually there's like additional proteins, like these little CCR5 and CXCR4 proteins that the virus likes to also make contact with before it actually triggers the entry. And when it triggers the entry, it's the stem part of the spike that kind of like pokes through, kind of drills like a hole through, latches on, so that the virus can eventually fuse and meld in, and the capsid can finally enter. So we said there's a strong interest in trying to look at those people, those special people that have that CCR5 mutation, and maybe that might help us come up with like an interesting angle for a preventative or possible cure. But I'll stop there. I'll see you guys tomorrow in lab.